Okay, so our topic today is going to be monoclonal antibodies. Here contains your specification. So this will show you all the things they can test you on in terms of what they do. This has come direct from the AQA specification. So this is the content you need to know. So we're going to look at the production of monoclonal antibodies and then more importantly looking at the uses. So three things I just want to focus on in today's session are the production of monoclonal antibodies. So how are they produced for human gain? They are produced naturally, which I will talk about in a second, but we're talking more about a medicinal use of monoclonal antibodies. How are they used? So there's lots of different uses of them that we will talk about, and I'm going to show you in a bit of detail, in particular, how a COVID test and a pregnancy test work, because that's an application question. And then you could get a 6-mark question to evaluate the uses or evaluate monoclonal antibodies. So again, I will show you how I will structure that answer, which will enable us to get 6 out of 6 in a 6-mark question on evaluation. So before I actually talk about monoclonal antibodies, I'm just going to talk very generally about an antibody. So an antibody you should be aware of before. The key thing to talk about when thinking about antibodies is every single antibody is a protein. Okay? So if the antibody is a protein, that should instantly be telling you a couple of things. A couple of things to relate to when thinking about the fact it's a protein. Well, you should instantly be linked to the fact that obviously if it is a protein, then the way in which an antibody is produced is via the process of protein synthesis, which I will cover at some point, but it's a level 2 paper, paper 2 topic, so we'll do that a bit later. And then again, protein synthesis, two things that should come into your mind when talking about it are the first stage of protein synthesis, which is transcription, and the second stage of protein synthesis, which is translation. The question could ask you, if a cell produces lots and lots of antibodies, how is that cell adapted for its function? Well, all protein synthesis occurs in a ribosome. So any cell that produces a protein will have lots of ribosomes. In particular, a cell that produces antibodies will have lots of ribosomes. And you'll also need to release lots of energy in order to produce those proteins. So that cell will have lots and lots of mitochondria. So that's just an application question. So you know what cells look like, but this is now linking it specifically to the function of proteins. So these proteins then are produced by a white blood cell. So one of the functions of your immune system is to produce antibodies. To be more specific, so a higher level answer than just saying a white blood cell, you actually need to know specifically for this bit of content the type of white blood cell that produces an antibody. And this is called a lymphocyte. So lymphocytes produce white blood cells, okay? Antibodies look like this. In its simplest form, that is roughly what an antibody looks like. So if I have a cell, this could be any cell, this could be one of my cells, but more likely we're talking about a foreign cell like a pathogen. Every single cell on the surface of the cell has these tiny little proteins. These are called antigens. They are unique to that organism, okay? So the antigen is unique to the common cold. It's unique to Ocean. It's unique to Tifa. It's unique to Mr. Duffy. The antigen is unique to that organism, okay? It's not even species related. It's unique to that organism, okay? This antibody, or the antibodies that are produced, the aim of the antibody is that they are specific, unique to that antigen. But the word I'd like you to use rather than specific or unique is we say that the antibody is complementary. Okay? It has a specific shape. So the antibody, to be more precise, this part of the antibody, this very top part of the antibody, has a complementary shape to the antigen. So it binds to that antigen and then it does lots of different functions in ways in which it can help your immune system. Okay, so that's antibodies in general. We're now going to focus more on actually specific antibodies, which are called monoclonal antibodies. So in terms of monoclonal antibodies, I just want to talk about a definition first. So if we break the word down, so mono obviously means one, okay? Clonal means a genetically identical copy. So a monoclonal antibody is literally lots and lots of copies of the same antibody. So clones of the same antibody, which means they all have the same shape. They are all complementary to the same antigen. So that's what it means. They are complementary to the same antigen. So complementary to same antigen. Okay, so that's our definition in terms of a monoclonal antibody. And they are produced by lymphocytes, as we said here. So we're now going to talk about the artificial production of monoclonal antibodies. Okay, so say I want to produce a monoclonal antibody against a particular disease. So the disease we might choose might be something like HIV. Okay, so we want to have monoclonal antibodies against the HIV disease. Well, the first thing I'm going to do is I need to have the thing I want to make monoclonal antibodies against. So I've got HIV. So it could just be the whole virus itself. It could just be the HIV antigen, but I have to have the antigen. So if I was talking about how to make monoclonal antibodies against a particular drug, that's also fine. I just need to have the antigen for that particular drug. So the first thing you do is inject that antigen into a mouse. Okay? That mouse then, the mouse's lymphocytes are then going to produce complementary antibodies to the HIV antigen or the antigen that you are testing against. So the antibodies make those. Those lymphocytes then go to a particular organ in the body. They then go to the spleen. The spleen is the organ in the body that stores all your antibodies and stores lots of your white blood cells. So those lymphocytes get transported to the spleen. What we then do is we take out those lymphocytes from the spleen. So we remove those lymphocytes that have produced those complementary antibodies from the organisms, from the mouse's spleen. And what you then do is you combine it with a tumour cell. So this diagram here is representing a tumour cell. Okay? The word I'd like you to use rather than saying combine, and this tends to be the word they use in a mic scheme, is they fuse together. So I fuse a tumour cell and a lymphocyte together. What I then create, once I fuse them together, and this is always on a mark scheme, I create something called a hybridoma. So that's the combination of the lymphocyte and the tumour cell. It's called a hybridoma. The reason we've combined it with the cancer cells, because we know cancer cells divide uncontrollably. So this hybridoma is now going to divide and 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 divide. And I'm going to have lots and lots of these hybridomas produced. And these hybridomas all produce the complementary antibody. So all I then have to do is I then just have to purify this solution to separate the antibodies from the hybridomas. And I've now got a solution filled with complementary antibodies to the antigen that was injected in the first place.
Sorry, do you always combine, are the lymphocytes always found in the spleen?
Yeah, we always take them from the spleen. The spleen is the easiest place to remove them from. It's quite external from the body, so you can get to it quite easy. It also, if you were to damage it, it doesn't have that much of a significant effect on the mouse. Yeah.
Do you need to know how they are purified?
No, you just need to know they get purified to separate from the hybridoma from the antibodies. We want the pure antibodies. Okay, couple of kind of higher level points I just want to note here. Okay, first of all, how do they divide from one hybridoma cell to lots of cells? Well, hopefully, the fact that it's dividing, you should all be thinking straight away they divide by mitosis. So just be aware of that. It's another example of mitosis, that hybridoma, because we're making identical copies. Mitosis is the genetically identical copy. Next question, which would be a really high level question, more like an A-level question, is why did we choose to use a mouse? Okay, mouse has got lots of positives as to why we use them. The first reason, rather than using, say, something like a fly to do it, we use a mouse because a mouse is a mammal. So that means a large majority, 90% plus of the mammal's genome is going to be similar to a human, so we assume the effect would be the same. Equally though, the reason we use a mouse and not a rabbit, a dog, all of those things, an elephant, is mice are very small, which means they are very easy to manage in a lab. So you wouldn't do an elephant because you can't have thousands of elephants in a lab. You can have thousands of mice in a lab. They reproduce really quickly, so I can get lots and lots of those. So that's the reason we would use a mouse for this monoclonal antibody production. Okay? So I've now got lots and lots of complementary antibodies against a particular antigen. So we're now going to talk about specific uses for that. So all of you in your life have used monoclonal antibodies because the original and the simplest COVID tests use monoclonal antibodies. They use those to detect whether you in your body had a particular antigen, i.e. you had the COVID antigen. So that's one example you're all aware of. I'm just going to now go into a bit of detail as to how they work. So this example here is kind of a simplified version of how a monoclonal antibody works. This could be a COVID test. So they could talk to you about COVID tests, but I'd say more likely they'll talk to you about pregnancy tests. So one of the first examples of how monoclonal antibodies are used is in a pregnancy test. Okay? So this is what your monoclonal antibody test looks like. And this, you could also use this for drug tests, actually, which I'll talk about in a second. Effectively, this allows you to detect the presence of any antigen. So it could be COVID, it could be pregnancy, but it could be steroids, it could be nicotine, it could be testosterone levels. This could be exactly the same for all of them, okay? This bit here is where my sample goes. So for a pregnancy test, obviously it'd be a woman urinating onto that part. For you, when you do the COVID test, it could be your saliva, it could be your mucus, it could be a blood test. So this is where you put your sample that you want to see if the antigen is present. Inside here then, I have got lots of these things here, which are of course my antibodies. They are complementary antibodies to the antigen I'm trying to test. So these are my monoclonal antibodies. In addition then, attached to these antibodies, in your specification, it often says it's just a blue dye. And if you think of it as that, that's fine, because in its simplest form, it is just a blue dye. I would say, if we were to talk in a bit higher level about what it actually is, more often than not, it's actually an enzyme. And that enzyme changes the colour from colourless to often blue. But the simplified version is it is a blue dye. So the sample gets put onto there, and if that sample is present, or even if it's not present, it will then bind to my complementary antibody like that. Okay? I've then got my test strip. So here I now have another set of antibodies. These are also complementary to the antigen. So the same as this, they are also complementary to the antigen. So if that antigen is present in the sample, it will then bind to it like this, and you'll end up having something that looks like this. So all I've actually done is one of these is the same as one of those. It's now just joined together to make this. And that then means it shows up blue. Okay? So if the antigen is present, it will bind to this complementary antibody on the test strip, and then it will go blue. Yeah.
The ones in the test strip are fixed on place, right? But
They're fixed, sorry, yeah.
And then, but is it the antibodies that are on the sample that carry the antigen?
Yeah, these ones are free. Good point.
Do those ones carry the antigen?
They carry the antigen, yes. So they are freely moved because the antigens should come from the sample. So they then bind to this bit here. If the antigen is not present in the sample, they will not bind to this bit here. So this one should not go blue. It will only go blue if the antigen is present. But you also have a second type of monoclonal antibody in the second test strip. This is called the control strip. This is looking to see whether your test strip is actually valid, whether it is a legitimate test or not. And this one is complementary to the complementary antibody. So it's not the antigen, it is complementary to this original antibody, this one here. So it will always bind on there because that one will always be freely moving from the first sample. So that will always move along, and because it's got the blue dye on it, or the enzyme which changes the colour blue, this will show you whether the test works or not. So you should, a positive test result would be 2 blue lines. A negative test result is 1 blue line on the second site. If you get no blue lines, that does not mean you do not have the antigen. No blue lines means a faulty test. So no means a faulty test, which is why a woman sometimes ends up spending about £30 on 3 different pregnancy tests because either they don't like their answer or they got a faulty test. So that is how it works. And I've seen a question ask you to describe how something like a pregnancy test or a COVID test works. Okay? So that's very common use of them, COVID tests and pregnancy tests then. Talking about more general, monoclonal antibodies are used to detect the presence of a substance. Most common examples of a substance we're looking to see are present are a pathogen. So the example we've just spoken about is COVID. The example we spoke about earlier could be HIV, but it could be any pathogen we're talking about. For your exam specification, they could link it to measles, gonorrhea, salmonella, malaria, and HIV. You could do it for TMV and rose black spot. Of course, they're plant diseases, not human diseases. Okay? You could be testing for the presence of a hormone. Okay? So that's how a pregnancy test works. You don't need to know this, but they could have an application question. The hormone that's found in pregnancy, or the hormone that's released in pregnancy, is something called HCG, human chorionic gonadotrophin, and that's what a pregnancy test tests for. And in sports, monoclonal antibodies are used to test for the presence of a particular drug. Monoclonal antibodies have got excellent now. Not only do they detect whether it is present, they can also now tell you the quantity. So as well as just telling you if it's there or not, they can now tell you how much of it's there. So all of us in our body would have testosterone. So we wouldn't just want to test whether it's there or not, because it should be there. But if you're a drug test in the Olympics or something, you want to see, is the amount of testosterone in the body a fair limit? So now we can use monoclonal antibodies to test the quantity that is there, as well as just the presence of it. Okay? So that's the main use of it. You could use it in food testing as well to see if a particular food is present in an organism. The best use and kind of most recent use of it is in cancer treatment. Sometimes this is called immunotherapy. Immunotherapy is just the use of monoclonal antibodies to test cancer. So hopefully you're all aware that historically, the 2 main treatments for cancer are chemotherapy and radiotherapy. As much as chemotherapy and radiotherapy are incredibly effective, they target all cells. To be slightly more specific, they target cells that divide quickly, but you inevitably kill lots of healthy cells during chemotherapy and radiotherapy. So for example, something like nail, skin, hair, which are cells that divide quickly in your body, that's why they get destroyed in chemotherapy and radiotherapy, because they're dividing quickly. And the chemotherapy drug and radiotherapy drug cannot tell the difference between a skin cell and a cancer cell. That is where immunotherapy is so good. So what we can do is we can produce now monoclonal antibodies against cancer cells. So this is a monoclonal antibody that is complementary to cancer cells. So it could be a breast cancer cell, it could be a skin cancer cell, it could be a liver cancer cell. It could be any type of cancer cells we can now produce monoclonal antibodies against. But what we can also add to that monoclonal antibody is you can then add the drug. So you can add that chemotherapy drug specifically onto the antibody, which then means this is only going to bind to a cell if the cell has the cancer antigen. If it's a normal cell, your normal cell does not have the cancer antigen, so it's not going to bind to it. But this cancer cell has got the antigen, so the antibody will bind to it, and then it can just target those cancer cells. So it means you're not destroying lots and lots of healthy cells. The word they sometimes use to describe this type of treatment is a magic bullet. So just be aware of that kind of colloquial term to describe this process, because it is literally magic and it's killing the cells you want to destroy. So that's a monoclonal antibody cell used in immunotherapy. So that's one of the best uses of it now. Final thing I want to do is just talk about how you evaluate it, okay? So this could be a 6-mark question asking you to evaluate it. It could just be a 2-mark question which says give 2 advantages of monoclonal antibodies, or 2-mark question, give 2 disadvantages. But I'm going to prepare you if it was a 6-mark question to evaluate the whole of monoclonal antibodies. Any evaluate question, if they are levelled, so level 1, level 2, level 3. Level 1 is 1 mark or 2 mark. Level 3 is 3 marks or 4 marks. Level 3 is 5 marks or 6 marks. All of us in here, of course, should be aiming for the level 3. In order to get the sixth mark now, you have to give a judgment. Okay? So that's the difference now between getting a 5 out of 6 and 6 out of 6. You give a judgment. So what I would encourage all of you to do, whenever you answer an evaluate question, is structure it really simply how I'm going to structure it now. So I would just start by saying, advantages. So what are the advantages of monoclonal antibodies? Well, as we know, monoclonal antibodies are specific. So they only target a particular antigen. The advantages really we're talking about are the uses. So monoclonal antibodies can use to detect the presence and quantity of a substance. And the other excellent use or advantage of them is in cancer treatment. But there are disadvantages. First disadvantage is there are lots of ethical considerations with monoclonal antibodies. The first one, as we spoke about earlier, is in order to produce monoclonal antibodies, we have to do some animal testing. And there are ethical considerations with animal testing because we are affecting the life of a mouse. Equally, we are combining your lymphocyte with a cancer cell to produce my hybridoma. That could lead to significant side effects. And there have been incidences of people who've had monoclonal antibodies who end up with cancer. So there's a particular case called TGN1412. You don't need to know that, but that's a case that was in the news a few years ago where people died from their testing of monoclonal antibodies. Okay? It is also expensive. So that would get me 5 marks if I just wrote it. And even if I wrote it like that, and I'd encourage you to separate it into advantages, disadvantages, and you can bullet point it. But then in order to get my 6th mark, I would then say, but to conclude, the benefits outweigh the negatives. And that would then get me 6 marks. You don't even have to say anything massive, but it's just giving a statement. So that's then all the things we want to cover. In terms of past papers, what I'd encourage you to do, as I said before, is re-watch this. When you get to this part, pause it, have a go at that question, and then look at the mark scheme. Have a go at this question. This is very much an application question about HIV. And here's the mark scheme. And here's another question here which is specific about fluorescence. So pause it. Here's a mark scheme. And there's one final one which talks specifically about how does a pregnancy test work. So if you just want to test your understanding of how those work, have a go at that question. Pause it. And then there's the mark scheme there.